Well, it's really also my great pleasure to be here today, and I hope to interact with all of you in this rather unique setting. I think we probably also took took the sort of the task of presenting here very differently. And what I took upon myself is to actually try to convince you today that we are doing a fantastic job as plant scientists, but we're not always looking at the right level and maybe not at the right type of variation. And so as the title of my talk actually reveals, what I would like to emphasize today is that we're not protein-centered enough as plant biologists. And we can strongly disagree on that, and that's why this is a great setting. We can fight outside, we can talk inside, anyhow. I think we probably all realize that in the past years, decades, there has been an enormous revolution in what can be done in plant sciences. In plant sciences, when we talk about the genetics underlying plant development, plant traits, plant crop productivity, etc. I just took a couple of examples here where now with pangenomes, with single cell genomics, with population genetics, and also with precision genome editing, etc., and all of that, even in what would classically be non-models, there's an enormous sort of advance in what we understand and what we can achieve as plant scientists. So this is absolutely amazing. And when we go back to the sort of the central dogma that was formulated such a long time ago, I mean, this is more than 50 years now, this is something that we've all learned as we were studying, that of course DNA encodes through transcripts proteins that will eventually have to do something. And I think that the focus in plant biology has largely been on variation at the genome level. We just heard about GWAS analysis, QTLs. That is genomic variation between locally adapted cultivars or locally adapted ecotypes in this case. And of course, that will translate to changes in RNAs that will eventually have to culminate into some functional changes in what proteins do inside cells. Now, I would just like to tell you one little secret that we often tend to forget about this central dogma in molecular biology, and that is that variation in genes and in transcript rarely correlates to variation in proteins. This is a paper from, I think last year, maybe this year, that took this test in a single-celled organism, in a yeast, to ask within a very large panel of natural isolates, in this case more than 1,000 natural isolates, let's measure transcript levels for every gene, and let's measure protein levels for every gene. This is what most correlations look like. The transcript levels go up and the protein levels don't. There's a complete lack of correlation. So this is across all the genes and proteins in yeast, the level of correlation. There's very few proteins that correlate positively with RNA transcripts, but most are either neutral or even anti-correlated. So in other words, it's fantastic to know single cell transcriptomes, but just don't expect that you'll see the same level of variation at the level of proteins. Right? So please keep this in mind, at least for the rest of this talk. Now, so what I would argue is that in the past decades, there has been a very large emphasis on coding variation, but there has been much less emphasis on what that actually means for cells. So for example, with those proteins and their enormous multidimensionality of function, there's very little that we actually know about protein quantities in cells, about where proteins are in cells globally, about their post-translational modifications, their interactions, and for example, their homeostasis. So we just heard about the N-enfold pathway that is selectively degrading proteins in an oxygen-dependent manner, just as one of probably thousands of mechanisms that are selectively controlling levels of proteins. So again, what I would like to do in this talk is to really brush over a number of examples from the work in my own lab to demonstrate to you how a focus on proteins can actually lead to insights that would otherwise be very hard to attain. And it's not going to be very deep, but I'm very, very keen on discussing that with you more in the days to come. Now, so what I would like to do is to basically tackle two topics here. So I would like to first focus on protein networks and spatial proteomics. So that's about protein interactions and where proteins are inside cells. And then lastly, I would like to focus on a couple of examples that are focused on signaling. signal processing, signal perception, mostly with an emphasis on having the correct speed, the correct kinetics of experiments that actually match the kinetics of the signals that plant cells are responding to. All right, so in my team, we're very interested in globally multicellular development in plants. So we're trying to understand how individual cells are actually giving rise to functional multicellular assemblies of cells through, for example, oriented cell divisions, giving rise to morphologies, not so much cell adhesion, through cell specialization after division, and also through establishing polarities at the level of individual cells that are guiding asymmetries. Now, apart from that, we're very interested in signals that are controlling this. Auxin is one that we've been studying for decades now, and more recently we started to study, for example, mechanical signals, and I will get back to that at the very end of my talk. So you're going to see a bit of that during this talk, and what I would like to focus on first is really a vignette of our use of proteomics in understanding what I think is a very fundamental process, namely the polarization of individual cells. How do cells establish one phase that is different from the other phases of the cell, and then use that information to guide, for example, the orientation of cell division, which is a prerequisite for morphogenesis. Now, if I sort of abstract this, what is actually happening is that in individual cells, there is essentially a manifestation of the axes that the cell finds itself in. So in other words, if there's a multicellular structure here that has an upside and a downside and an outside and an inside, etc., individual cells need to be instructed of those coordinates so that they can align themselves with these coordinates. After all, cell division is by definition a local genetic program that can be guided by extracellular information, but it's locally executed. So how do cells actually lay down information at the individual cell level that corresponds to supracellular axis? That for me is the essence of understanding cell polarity. And that's something that in plants we don't know all that much about, if we're modest. Now, so some years ago, we made a little step forward in understanding cell polarity by identifying a group of proteins that we call soseki, which are localizing towards one of the edges in the cell. So this is the upper and outer edge of cells in the Arabidopsis embryo here. I'm not going to discuss any details here, but the important thing is that these proteins are polymers. Here you see the crystal structure of one of them. They're polymers, and they assemble as polymers in the edge of the cell, and thereby allow other proteins to go to that place in the cell. By having a very high local concentration, they can overcome low affinity. So that's avidity instead of affinity. And as polymers, they can therefore act as sort of a scaffold in helping the cell organize itself. Now, we have a research program on this, and I'd be happy to discuss it in detail. But today I would only like to show you how we're using proteomics approaches to use this as a starting point to better understand polarization of plant cells. So this started some years ago with, I would say, a rather classical approach. We have this soseki protein that you see here on the screen. It wasn't really known what it was doing. It wasn't really known what context it operates in. So the strategy we took was to use a mass spectrometry, affinity purification mass spectrometry approach to identify other proteins that are associated with it. Here you see one of them. SIB stands for Soseki interacting BTB. It doesn't really matter. Bottom line is you can identify other polar proteins. Here you see this one is also laterally polarized. So we did the trick again, right? Using this SIB1, and then we identify yet other polar proteins. And we could have continued this for, I don't know, an eternity and keep identifying new polar proteins to add to this sort of growing repertoire of proteins that are polarized within cells. We felt that it might not be very smart to keep doing this again and again and again, because it would be extremely resource consuming and not very effective. Instead, we thought it might be a good idea to do what we would call systems proteomics. You can give it any name you want, but the bottom line here, and this is work from Victor, Zhenya, and Mark in the lab, is that we essentially collected lots of these polar proteins through the means that I just showed you in the previous slide, and then connected each and every one of them to a proximity ligation enzyme that is called TurboID. And what that does is it converts biotin into a chemically active version of biotin that will locally within the range of 10 to 20 nanometers attach to lysines. So in other words, non-selectively, any protein that is in the vicinity of this protein here will be covalently labeled with biotin. We can isolate it and identify it by mass spectrometry. And we did this for a whole panel of polar proteins that are either on the lateral side, on the top side, in the edges, et cetera, plus some controls. Now, this was an enormous experiment. Well, I guess about 200 or so mass spec runs. And what you get out of this is something like this. And I guess maybe the systems biologist gets very excited by this. Here we have 25 clusters of proteins that together resemble about 3,500 proteins that are enriched in one or more of these experiments with one or more of those baits. So this is a massive data set, and I cannot read anything out of this. It's extremely hard to get any knowledge out of this. You might go and see, well, there's a cluster of proteins that are enriched in one or the other baits, and maybe it's localized there. But when we did this, we realized there's something really important that we're missing in plant biology, and that is systematic insight into where proteins are in cells. In yeast, there's a collection of 6,000 strains. With GFP, you can look at all of those strains, and you know for every protein where it is in the cell. In plants, we've maybe localized 2,000 proteins or so, maybe 3,000. And those are the proteins for which we know localization. But the other 27,000 proteins in Arabidopsis, we don't know where they are. We have some predictions. And we felt that that is something that we need to know to interpret a map like this. So I was very lucky that at the time, a graduate student in the lab who had been trained in a proteomics lab, Monique, decided to take on this challenge to develop spatial proteomics for plants. And what I mean with spatial proteomics is not cellular proteomics, but it's actually subcellular spatial proteomics. Where are proteins within cells? And the method she used, I would say, is biochemically very classical. You break open the cell in a very careful manner, and then do a successive differential centrifugation steps. So first you spin a little bit, take the pellet, run the supernatant a little bit harder, and so on and so forth. And what you end up with are 10 different fractions here. And then we use the latest innovations in data-independent acquisition, a mass spec, to quantify all proteins in all of those fractions. And now every protein will have a unique profile across all of these fractions, but proteins that are in the same place in the cell will have the same profile, right? So we can use the same type of statistics that are used for single cell transcriptomics to identify clusters of proteins with the same localization. Of course, when we know some of those proteins and their localization, we can expand that prediction to neighboring proteins in the map. So this is what it looks like in reality. These are Arabidopsis roots, so we get a mapping, statistical mapping of all of the proteins that we can quantify. We can use statistical methods to identify clusters, then curate those based on known information. For example, all of these red ones here we know to be in the cytoplasm, so that is the cytoplasm cluster. And then eventually we get a map like this. Now, I should tell you that this is an experimental localization of 8,500 proteins in Arabidopsis roots. This is not a prediction, right? This is actual data, actual experiments. And now for all these proteins, we have a pretty good prediction of where they are supposed to be inside of cells within the root. We've done the same thing for whole seedlings. There we have about 6,000 proteins localized. And also for Mercantia thallus, there we have about 4,500 proteins localized. Okay, no details here. I just want to show you that when we take proteins there, and we did this for about 70 or 80 proteins, and we localize them experimentally, they are where they are supposed to be. Right? So it's an accurate prediction of where proteins actually are based on centrifugation. Now, importantly, when we look at protein complexes, you expect them to be in the same place, and that's what they are most of the time. I mean, of course, there are exceptions, but generally protein complexes tend to stick together, and that validates this map. Back to the proximity proteome that I showed you before, right? So this was proteins that are localized in roughly the same place based on the biotin modification with the turbo-ID enzyme. Now what we can do is we can take one of these clusters of proteins that are supposed to be in the same place, and this one is a little bit enriched in the lateral domain, and we can take all those proteins and map them onto the spatial proteome that we determined. And now we see that this cluster splits apart into what is a plasma membrane cluster here and an endosome cluster here. And very importantly, you see that some proteins are more closely connected in this spatial map than others. And that tells you something about how intimately they're connected in the cell. Just to guide you to how we are using this, we identified 3 enzymes here. They're called PAT, that stands for protein acyl transferase. Those are proteins that acylate target proteins to target them to the membrane. And it turns out that these enzymes are actually polarized themselves, so they're on the lateral side of the cell. And we more recently found, and I'm not going to discuss that in detail, that they are actually guiding the Soseki protein to the membrane. So through this method, we've been able to identify that the Soseki protein first goes to the membrane through a charge-based mechanism, but then there's this local acyl transferase that adds an acyl group to insert it in the membrane. And then lastly, polymerization ensures that it ends up in the corner. Right? And I'm telling this not to give you any details about the polarity system, but rather to show you that it's the combination of spatial proteomics and proximity proteomics that allowed us to identify this mechanism. I don't think we would have found it any other way. All right, good. So that was a sort of a vignette on spatial proteomics and proximity ligation. I would like to continue with post-translational modifications in the context of signaling. And here I would like to make sort of a point about speed. But let me first sort of tell you why I think that's relevant. I think in this room, we all agree that plants are absolutely masters of signaling. You can use any adjective you want, like sessile, some like it, some don't, or whatever other adjective. Plants need to sense their environment, and they need to respond in order to build their bodies, respond to the loss of water, and all of these things. And over the past decades, people have identified lots of receptors, lots of signaling components, lots of responses, and I think those only add to the notion that plants are extremely good at signaling and also integrating lots of signals. But I would like to point out one thing that I think, again, is a little bit of a skeleton in the closet of plant signaling. And that is that when people study signaling, this is very often at a time scale that I think is not really reflecting the biochemistry and biophysics of signaling systems. And why am I saying this? When we think about a receptor being bound by a ligand, inducing a complex at the membrane, for example, inducing protein phosphorylation, inducing adapter recruitment and or relay of the signal, and eventually transcription, then the first steps here are limited only by diffusion. And diffusion at short scales is doesn't take a very long time. So these first steps here are expected to happen within seconds at best. It's those steps here towards adapter recruitment, effector phosphorylation, transcription, that will take minutes, maybe hours in some cases. Now, if you look at the literature in plant signaling, we know quite a lot about what happens after minutes and hours. I would say hours more than minutes, but we don't really know that much about what happens within seconds. So I think there's a problem here, is that we're actually not seeing the initial events in signaling simply because of technical limitations. So what I'm going to advocate is that we need some technical innovations to actually see those first steps so we can learn more about signaling and about crosstalk. And I'll give examples of this. And the first example comes from our work on auxin biology. Now, when I say auxin, you will probably all agree that this is an important hormone that does lots of things to plants. And most of what we know about it is based on this system. This is a transcriptional system that uses transcription factors, they're called ARFs or auxin response factors, that uses repressors that are called auxase, and that uses a receptor that is called TIR1. So receptor leads to degradation, or binding to the receptor leads to degradation of the repressor, leads to activation of transcription. And this is something that we've been studying over the last years, actually, along with lots of other groups. And there's something that occurred to us when we started looking at evolution of the system. So first of all, the system is found in all land plants. That's very important. And there is a component that is also found in the algal sister group to land plants, but the entire system is not there. So in other words, algae have the transcription factor, but they cannot respond to the hormone in the same way. So that's one thing. Secondly, so those algae do respond to auxin, right? So they don't have the nuclear response system, but they do respond to auxin. And in addition to that, over the years, there have been many responses that have been recorded in land plants and also in algae that are simply too fast to be mediated by transcription. So if you have a change in membrane polarity within 1 minute, it's impossible to make a protein in that time. It's simply physically impossible with the rate of transcription, the rate of protein synthesis, folding, et cetera, et cetera, et cetera. So there has to be another system that can mediate rapid auxin responses and that can mediate the auxin responses in species that don't have the nuclear system. So we've postulated some time ago that there has to be a second unknown auxin response system that is rapid, that is conserved in land plants and algae, and that controls something that is also happening in land plants and algae, and that's not flower development, that's not root development, because algae don't make that. So it has to be something very basic, very fundamental. And we have started searching for sort of an intrinsically rapid system, namely a phosphorylation-based system, because in many other eukaryotic systems, those systems mediate rapid signaling. You can very rapidly switch between any two states of a protein by adding or removing a phosphate group, which adds charge, which adds the possibility for hydrogen bonds, et cetera, et cetera. And it's reversible. So kinases and phosphatases mediate this type of response. Now, we started looking for this in the context of auxin that had not really been explored very much at that time. There was work started by Mark Roosjen in the lab, and he sort of took it upon himself to push himself as far as he could to get the most rapid output. And this was initially done in roots, where he could push it as far as getting 30-second treatment, 1 minute, 2 minutes, and then took corresponding mock controls. And the bottom line was that there's a lot going on. There's about, let's say, 3,000 sites on about 2,000 proteins that are cumulatively affected during those first 10 minutes of auxin treatment. But very importantly, there's hundreds of proteins that are differentially phosphorylated within 30 seconds. This is all happening while you're waiting for transcription to occur. Right? So there is this apparently unseen response that is happening very rapidly when plants sort of encounter auxin. Now, importantly, when you have these time series with 30 seconds, 1 minute, et cetera, you can use simple regression logic to identify relationships between protein kinases and predicted substrates, because the kinase would always be active before the substrate gets phosphorylated. And you can predict the activity of any kinase in any organism because there is an activation loop in the protein. When that has a phosphate, it means that the kinase is active generally. And so what we have here are activation loop phosphorylation sites in a lot of kinases. Blue means up, yellow means down. And you see over the course of the experiment, the dynamics of activity of those different kinases. And then we can use, as I said, regression logic to couple those to the dynamics of phosphosites in the proteome. And that gives us a network where we can predict for any kinase how relevant it is for this response. So there's a bunch of kinases here on the top right that are predicted to be very important for this auxin-dependent phosphorylation. Keep this in mind. Because we also did this in other species to ask the question to see if this is an Arabidopsis-specific response or maybe something more that happens more broadly. So in the meantime, Paulette, André, and Sjoerd have done this in Arabidopsis, Viscomitrium, Marcantia, Benium, an alga, Klebsiella alga, Ceratopteris, a fern, and another alga, Cara. So, and the bottom line is, in all these species, we find the same response. Hundreds of phosphorylation events that are happening within 2 minutes in all of these different species. And very importantly, there's even a core set of proteins that is differentially phosphorylated in all these organisms, despite 800 million years of separation. Right? So I would say that's pretty impressive, that despite all of the differences that have also emerged, there is actually a core set of proteins that is targeted by auxin-dependent phosphorylation. Now, when we look at this core here, right, these proteins that are always phosphorylated, no matter what species you look at, there's actually a kinase there that I'm sure Christophe likes, because it's actually the group of kinases that he referred to, the RAF-like kinases. They are both very deeply conserved in their regulation by auxin, and they're predicted to be really important in mediating the responses in Arabidopsis. So we've looked at mutants both in Mercantia and in Arabidopsis. The plants are small. This is a septuple mutant that knocks out 7 of the B4 RAF kinases. In Mercantia, there is only one, and this is a single mutant. And the important thing is that when we look at auxin-dependent phosphorylation in these mutants, we see that both in Arabidopsis and in Mercantia, more than half of the sites are lost after an auxin treatment. So in other words, these are absolutely central kinases in mediating the phosphorylation changes in response to auxin across these 2 different species. And they were identified by phosphoproteomics. Now, what response do they connect? I mean, what physiological or cellular response do they mediate? We looked at several of these rapid responses that had been reported, and I'm going to show you data for this one, cytoplasmic streaming. So that is a turbidity that is created in cells by actin and myosin actions. So it's basically moving organelles around, so stirring the cytoplasm. That's a very ancient response in many eukaryotic cells. And auxin promotes this. This is here in Arabidopsis. This is in Mercantia. We've also seen it in algae in the meantime. Auxin increases the velocity of the streaming inside cells. And the mutant in the RAF-like kinase is absolutely insensitive to auxin, both in Arabidopsis and in Mercantia. So this rapid phosphorylation response is connected to a cellular response that is conserved across algae and land plants. So let me summarize this example here. What I showed you is that we've identified before that the nuclear auxin response is conserved in all land plants. That's present in all land plants. So it evolved around 450 million years ago. But what we've now found is that well before that, probably 800 million years or so ago, there already was another auxin response system, a rapid auxin response system that depends on the capacity to phosphorylate hundreds and hundreds, if not thousands and thousands of proteins in a very rapid time frame. Now, as I said, this is phosphorylation of phosphoproteomics-based identification of a new pathway that turns out to be very conserved. I cannot tell you what it's important for. But very recently, we found that when you look at these rapid signaling pathways, you actually also start to detect crosstalks and interactions that maybe are not so obvious. And this comes from work recently done in the lab by Jashu and Aaron. So Jashu comes from an immunity lab, and he got very interested in auxin for the very simple reason that when we look at the proteins that are targeted by this rapid auxin phosphorylation, we find that 60% of the proteins that are targeted by flagellin, that's an immune elicitor, or bacterial signal, 65% of those proteins are actually also targeted by auxin. If the plant detects that phosphoproteome, it might not even be able to tell whether it was auxin or whether it was a microbe that it encountered. And if you look at the types of proteins that are differentially phosphorylated, these are all hallmark PTI or pathogen-triggered immunity phosphorylations, and they're targeted by auxin. So we believe that there might actually be very intimate connections between this type of auxin signaling and immune signaling that we're trying to explore now. But again, that's not something that you would easily have predicted based on genetic data. Okay, so the last example that I would like to give you is more related to the speed aspect. I just told you that we found an unexpected new rapid signaling system to a hormone that has been studied for almost 100 years now, auxin. But that's maybe not the first molecule you think of when you think of speed. Instead, when we think of speed, let's skip this. Let's skip over this for now. When we think of speed and rapid signaling, I rather think of things like mechanics, where sort of a physical signal that is immediate has to be converted into a biochemical output in cells. So there has to be some kind of conversion of a physical to a chemical, a biochemical signal. And that, in that case, does not require any diffusion. It doesn't require any biochemical conversion. That is really an immediate signal, a physical signal. Now, in animals, there's quite a lot known about how mechanical signals are perceived, and that often includes protein kinases that are activated by mechanical signals. So we felt that perhaps with these, if we think about rapid signal conversion, maybe mechanics is actually a very good test case for identifying such changes. Now, of course, there's a problem. I told you that with auxin, we went sort of out on a limb to get a phosphoproteome after 30 seconds. 30 seconds for a mechanical signal is an eternity, centuries. It's far, 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 far too long after the perception of the signal. So we need something that is a lot faster. And that is something that we felt we needed to invest in because we need different model systems for this. And we eventually ended up working with cells that have been established at the PSPD Institute in Ghent, so-called PSPD cells, because those are actually plant cells that are little clumps, but they have a wall. Here in pink, you see the cell wall. In yellow, you see the polarized Soseki proteins, so they're polarized. They have a wall, and they can respond to lots and lots of signals. Here we try ELF18, FLAG22, brasinolite, auxin. This is a 1-minute phosphoproteome, right? So they can respond to all of those signals. They have a wall, they're polarized. So we look at these as sort of plant HeLa cells, except that they have a wall. And maybe a nice cellular system to start probing rapid responses, because the nice thing about these cells is that you can pass them through a microfluidic device. That's not possible with a whole seedling or a root, but with these little clumps you can. So Ping Yosapo, he developed a device some years ago where he can pass little clumps of Arabidopsis cells through little constrictions to give them a mechanical compression, a mechanical signal. And now, depending on the length of tubing that we use after the constriction, say, for example, with 1 meter or 2 meters or 10 meters, we can change the timing after the constriction before we sample the cells. Yep. So now it's sort of a standing wave of cells that is continuously passing through constriction, and then depending on the tube, we can harvest after 1 minute or maybe 1 second, right? Depending on the tube. So I'm going to show you 2 examples here. One is a transcriptome experiment. We give 1 little compression and then measure what happens to the cell transcriptome. And the bottom line is that these, on the principal component analysis, all separate by color, which means the time, but not by treatment. So even after 60 minutes, we don't see the control and the treatment go apart. So in other words, we don't see a transcriptional response to this 1 little compression. Maybe that makes sense. Maybe you don't want to respond transcriptionally to something that is just touching you. What about phosphoproteome? Here we took phosphoproteomes after 5 seconds, after 15 seconds, 30 seconds, 1 minute, 5 minutes, and then we look at differential phosphorylation. And now we were shocked because there's hundreds of proteins that are phosphorylated after 5 seconds, but not anymore after 15 seconds. And then there's 100 or so other proteins that are phosphorylated after 15 seconds, but not after 30 seconds. So in other words, there's a wave of phosphorylation going through the cell that you can only detect if you look at 5 seconds or at 15 or at 30 seconds. And so that would basically be completely invisible to genetics. It would be completely invisible to any other means. And it's certainly not visible if you wait 15 minutes or 30 minutes to take a phosphoproteome. So what I'm trying to tell you is that there's probably a whole universe of plant signaling and plant sensing mechanisms that we're just not seeing because we're not using the right technology for it. Now, in this particular case, Cecilia and Isaac are working on 1 kinase that we identified here. We call it JEDI. The kinase is active after 5 seconds, but not after 15 seconds. It turns out to have a carbohydrate binding extracellular domain and a microtubule binding domain integrated in an intracellular kinase domain. This is localized at the plasma membrane in root cells. And it's a member of a large family, but even a single mutant in this 1 JEDI, it makes the root a little bit shorter, and it makes it a little bit less sensitive to an osmotic treatment. So I believe that what we have here might actually be mechanically activated receptor-like kinase, maybe mechanism implants. Okay, so I think I made my point clear that what we try to do is use the advances in proteomics and in protein biochemistry to actually fill in gaps that are necessarily present in plant biology, simply because there are things that you can only see when you look with those eyes. Good. So I think I've mentioned the people involved in this work along the way, including the alumni that have been involved in this. And I would be very happy to discuss this with you in the next days. Thank you.
Yeah, thanks.